Okay, let's try and unpack this for you. Think about proteins, those amazing little molecular machines in us. Now, what if you change just one tiny piece, one amino acid, or maybe several pieces all at once? All of a sudden, maybe that machine just stops working. Or even worse, it starts causing diseases. Predicting how these little tweaks affect a protein's stability, basically, how robust it is, well, that's super important. Critical, really, if you're trying to build new proteins or maybe develop drugs or just understand our genetics better.
Yeah, it's a really big challenge. Um, Imagine trying to figure out how swapping out one bolt or maybe a few different parts affects a whole bridge's structure. Even tiny changes can have these massive unexpected effects. And doing it in the lab experimentally, it's just too slow, too expensive to check everything. That's where, computers come in, these computational tools.
Right. And the material you shared today gets into a really interesting new tool for exactly this problem. It's called ddgemb.
That's it, ddgemb. So yeah, our mission for this deep dive is basically to figure out what ddgemb is, how it works its magic using just protein sequences, and what makes it stand out, especially on the trickier stuff.
Okay, so starting with the name, ddgemb, that M part
hmm
that suggests embeddings.
Exactly, spot on. ddgemb relies heavily on embeddings from these advanced protein language models. Specifically, the source mentions ESM2, which is pretty highly regarded. Think of it like, those large language models trained on Wikipedia or whatever. These are similar, but they're trained on tons and tons of protein sequences. They basically learn the language of proteins, how amino acids fit together, how sequences tend to fold, what it means for function, stability, all just from sequence data.
Huh. So it takes the protein sequence, the original one and the one with the mutation or mutations, and turns them into these rich number fingerprints, these embeddings.
Precisely. Yeah, it generates embeddings for the wild type, that's the original protein, and the variant, the mutated one. And then the really clever part is it calculates the difference between those two sets of embeddings. This difference matrix, it sort of captures the essence of the change caused by the mutation or mutations, structurally, functionally.
Okay, this is where it gets cool. So what does it do with that difference information, that sort of change fingerprint?
Right. Well, it feeds that difference matrix into a specially designed deep learning network. The architecture uses a transformer encoder, similar tech to what's in many of the big language models you hear about. Transformers are just really good at understanding context, how different parts of a sequence relate, even if they're far apart. And that's crucial because, a change here might affect stability way over there, or maybe multiple changes interact in complex ways.
Uh-huh.
Scientists call that epistasis. So the transformer processes all this change info to predict the AG value. That's the change in Gibbs free energy. It's the standard way researchers measure how a mutation impacts stability. More negative means less stable.
Got it. Okay, so it's pulling together some pretty cutting-edge AI protein language models, transformers, all to predict stability just from the sequence, and the source material really emphasizes its ability to handle multi-point variations. That sounds a lot harder than just one change.
Oh, it absolutely is, yeah. Predicting the combined effect of, say, 3 or 4 changes happening together, that's a major hurdle. Most tools out there, especially the sequence-only ones, they're really tuned for just single mutations.
So how did they actually test DGEM, put it through its paces?
So they mainly trained it on a big, high-quality data set of single mutations. It's called S2450, pretty standard, reliable experimental data. They were careful about the training, used rigorous methods to avoid bias. Then they tested it on separate, standard benchmark data
And
sets.
the results, how did it do if I needed a tool?
For single-point changes, tested on another benchmark called S669, ddgemb performed, right up there. State-of-the-art. It got top scores on things like Pearson's correlation, how well predictions match experiments, and had the lowest errors compared to, well, a whole bunch of other tools, both sequence-based and ones needing 3D structures.
Okay, top tier for singles. But what about that multi-point problem, the really tough one?
Yeah, this is maybe the most surprising bit, the most impactful result in the sources. They tested it on this specific, challenging data set for multi-point variations, PT-ML-NR. And ddgemb just significantly outperformed other methods, even ones designed for multiple changes like FoldX or Maestro. On this tough data set, ddgemb got a Pearson correlation of 0.59, which, shows a much better link to the real experimental results compared to FoldX, which is down at 0.36 on the same data.
Wait, hang on. So it was trained mostly on single changes, but it did way better predicting the effect of multiple changes. That feels kind of backwards.
Yeah.
But wow, really powerful.
It's fascinating, isn't it? It really suggests that the whole approach how the language model embeddings capture that detailed change fingerprint, and how the transformer process it seems to have this built-in ability to generalize. It's like it learned not just about single changes, but also something fundamental about how changes interact, those epistatic effects we mentioned, even without seeing tons of examples of multiple mutations during training.
Wow. That generalization is huge. So, okay, bottom line for you, the listener, who might actually need a tool like this.
Right. So for you, it means there's this new state-of-the-art method out there. It's great for single mutations, yes, but it also shows this kind of breakthrough performance on the much harder multi-point problem. And crucially, it's accessible. The researchers put ddgemb up as a web server.
Mm-hmm.
So you can basically just paste in your protein sequences and the variations, even whole lists of them, and get these stability predictions back pretty easily.
That's fantastic news for researchers. Okay, so quick recap. ddgemb cutting-edge tool uses protein language models and transformers, sequence only, nails state-of-the-art for single-point variations, and the big news, significantly outperforms others on multi-point variations, even though it was mainly trained on singles. Plus, it's available online.
That sums it up perfectly. Yeah, it's a really significant step forward.
So here's something to think about for you listening. How could a tool like this, one that can actually predict the combined effects of multiple changes pretty accurately, how could that really speed things up? Think about designing brand new proteins, maybe for industry or medicine, or trying to understand complex diseases where it's often not just one gene change, but maybe several acting together. Definitely something to chew on.